After talking about how proteins are made, you may come to understand that the formation of a protein is a very complex process. Hence, there might be some mistakes while the cells are carrying out these processes. Mutation is a sudden and permanent change in the DNA of an organism. Depending on the level at which mutations occur, they can be classified as gene mutation and chromosome mutations. Let's first look at gene mutations. The changes in the base sequence of the DNA is a gene. They can be brought about in a number of ways, such as deletion, insertion, substitution, and inversion of bases in the DNA. When certain changes are made to the base sequence, the reading frame will be shifted. For example, if one base or multiple bases that are not a multiple of 3 got deleted or inserted, like the example shown here, the reading frame will be shifted and the amino acid sequence will be changed. The proteins that are produced as a result of these changes are usually not functional. However, when it comes to the types of gene mutations, one base in the triplet code without shifting the reading frame, such as substitution and inversion, it may result in one different amino acid in the amino acid sequence. The phenotype of this amino acid sequence may be affected if it's altered sufficiently. Sickle cell anemia is a very common example when we are talking about mutations. Let's look at it together here. With base T being replaced by base A at one position, the polypeptide made from the base sequence has a different amino acid. As this type of mutation involves a base being replaced by another base, it is being regarded as substitution. Although the change in the sequence may not seem significant, it greatly affects the shape and subsequently the properties of the protein. The abnormal hemoglobin molecule form long fibers, causing the red blood cell to become sickle-shaped when oxygen levels are low. And when that happens, blood vessels will be blocked and cause severe risk to the patient. After talking about gene mutations, let's look at chromosome mutations. By definition, chromosome mutations are characterized by changes in the structure or number of chromosomes. So how does that occur? Well, when an error occurs during the crossover of chromosomes during meiosis, the structure of a chromosome may be altered. It can be brought about either by deletion, duplication, inversion, or translocation. Since there are 4 types of chromosome mutations, let's look at them one by one. A loss of genes can be caused by a segment of chromosome being lost, and this is called deletion. On the contrary, a gain of genes can also be caused when a segment of chromosome is repeated, and this is called duplication. When a broken segment of chromosome is reinserted into the chromosome in the wrong direction, the order of the genes on the chromosome is reversed, and it is called inversion. When segments of chromosomes are exchanged between non-homologous chromosomes and some of the genes are being exchanged between 2 chromosomes, it is called translocation. When there is a change in the chromosome numbers, it may affect the person's overall well-being. For example, people with Down syndrome have an extra chromosome 21 in their body cells. Normally, there are only 46 chromosomes in each of our body cells. However, there are 47 chromosomes in each of the patient's body cell. So how does chromosome mutation occur? During meiotic cell division, homologous chromosomes of chromosome 21 are not able to separate into two. They go to the same daughter cell. Therefore, some of the gametes produced have two chromosome 21, while some are missing chromosome 21 altogether. If the abnormal gamete contains two chromosome 21 fuses with a normal gamete, 3 chromosome 21 will be found in the zygote. Hence, the baby will be born with Down syndrome, with features such as poor muscle tone and flattened facial profile. You might wonder what causes such mutations. Well, it can occur either naturally or randomly, or being induced by mutagens. For mutations that occur naturally and randomly, they are called spontaneous mutations. And for mutation that are induced by mutagens, they are called induced mutations. For example, nitrous acid can change the base of a DNA molecule into another, while X-rays can ionize molecules to form free radicals. Free radicals are highly reactive and can cause damage to the DNA molecule. While most mutagens are usually harmful, with diseases or death as the result, it can also have no effect on the organisms or even be beneficial. Since some amino acids can be coded by different base sequences, mutations may produce a different code for the same amino acid. And sometimes, even with changes in the amino acid, it may not change the shape of the protein, and therefore no change to the function. Moreover, with alleles existing in pairs, although there might be mutation in one of the two alleles, it may be masked by the other allele that is normal. Mutations also provides a source of variation in a population and may give rise to characteristics that are favorable for survival in a certain environment. These variations are important for evolution. The taxonomical classification of the 3 mitten crabs has been historically controversial. They were once classified as a single species, E. japonica. However, previously E. sinensis and E. japonica were considered two distinct species. E. japonicus was first defined as a subspecies of E. japonica, but later classified as an independent species. Recent studies using different molecular markers have clearly shown that E. japonicus is a distinct mitten crab taxon. Not only does the mutation happen in humans, it is very common among all organisms, including crabs. A crab with an extra claw was found off the coast of Cornwall in the United Kingdom in 2013. It is relatively unusual to come across crustaceans with extra claws. Although it is unclear how the crab developed its extra pincer, it is likely that it was due to a random genetic mutation. This is the end of the video. Thank you for watching. Please give us a like and subscribe to our channel. We'll see you next time. Bye bye.